Meeting is being recorded.
All right. So we got a couple people in the waiting room. We'll let them in. And here we go. Gonna also. All right. So I am going to share my screen and get going on looking at iNaturalist stuff for our pre-meeting chit-chat / iNaturalist section. So usually this is when I would be really highlighting things from our club project where we have our 2026 virtual foray on iNaturalist. And there's a lot of gorgeous things to look at in here, including, just to plug my own observation, my friend Jamie and I found some tulostoma this past weekend, and that was a really exciting find.
Because tulostoma is a super weird fungus. It's like a stalked puffball, if you're not familiar. It's a pretty obscure genus of mushrooms that aren't well known, which is why it's cool.
But I was just chatting with Brennan about how I was curious if it's more on the underreported side because of how it's almost underground. Like these stalks appear underground. But you can dig them out, and sometimes I guess the sand, it's usually in sandy soil and it'll wear away. So just wanted to put it out there. Look for tulostoma, friends. And check out our project where you can become a member, and then your fungi observations will all automatically go into this project. Okay, so I would also usually be sharing the links to this, but I will let somebody else do that if they would like to. Okay, but I wanted to get started talking about some stuff that our speaker is going to be talking about when she's here and starts at 7 p.m. If our speaker arrives, Brennan, please let me know and interrupt me. And honestly, anyone interrupt me if you have something you want to highlight, because I would love to pick her brain about a lot of things to do with organisms, especially beetles associating with fungi and lichens. This is the other project I was really going to delve into. This is something that I made to follow up on my curiosity about organisms of any type that are not fungi, that are associating with fungi. So I've been especially using this for beetles, but anything could go here. It's supposed to be, and the project description kind of has the details, but it's supposed to be observations of the organism that's not the fungus. And then but you're showing it, it's actively showing the relationship, the interaction in question, so that can be evaluated. And that's why this is a traditional collection project on iNaturalist, as opposed to, like the ones where when you join this, if you join it and you observe a fungus and it's this within 2026, it will automatically get added to the project. You don't have to do anything. This one, you or somebody else manually adds your observation to the project. And I can demonstrate that for anyone who has any questions later. But really, I just wanted to get started showcasing some cool bugs that you can find and observe. So going in and looking at the observations. One of the fun things about curating this list is the more richness it gets, the more things that get added to it, and I really hope other people start adding to it, the more this sort of species rankings becomes informative. It's still not rigorous, but it could say, oh wow, like American carrion beetle, which normally feeds and is attracted to carrion. I didn't think of that as being particularly associated with fungi, but when we look at it, it's up there. Still low sample size, but and then you can actually go in here and explore like what fungi. I was just, saying that, okay, on stinkhorns, that is expected to me, but they've also been found at least twice on not stinkhorn fungus. I saw that Andrew was here, so I was maybe going to get an answer to this question live. Do you know what fungus this was that it was on? Because Beautiful up close observations. Oh, it's got a mite on it. Oh,
Oh,
whoa.
cool.
That's a mite type of thing on a beetle on a fungus. That's
I
great.
didn't take picture of the fungus. You're saying, right? Okay, well, maybe I didn't post it.
You may have taken a picture of the fungus. I have not been rigorous yet where I could look maybe at this date. Let's see, August 3rd, 2024. And I could go to your.
Yeah, I'm surprised that if I haven't, but sometimes I don't.
Yeah, you probably have. So I'm going to go in and search the exact date. I want 2024. I want August. And what was it, 4th? Let's test my memory. It's probably not great. Gonna go ahead and try 3rd. There we go, 3rd.
And Mariah, I do think potentially our speaker has joined. If you're there, Kristen, feel free to pop in and talk, because it's like an open forum kind of thing. You want to tell us more about bugs in the pre-meeting portion of this?
Hello. We were just talking about Necrophilia americana, the American carrion beetle, being something that has been seen on 5 different observations that I've put in here so far. And by put in here, I mean that I have a project that is specifically for organisms associating with fungi, including lichens, and it's like curated so people add their own observations or other people's that they see. One of my ways of doing this actually is once I figure out that something is being seen with fungi, sometimes I will go then just look through all of the observations of it and just scroll until I find one that is associating with a fungus. Like, okay, there we go. That looks probably fungal. Let's go. Yeah, that's a stinkhorn of some description. Yeah, column stinkhorn fungus. So to add it, I go in here and I search organisms. Because I've joined the project, it comes up for me pretty much right away. It's column stinkhorn that they're saying it was with. So we can add that. If I weren't sure, I could just put fungus. And then I have to hit this add to project. And then I can tell that it's on the project because it shows up right there. So now I would have another one. We could keep racking them up by scrolling through and seeing what we get. So one of the things I was really curious to ask our speaker about, if she's ready maybe to be asked.
We'll see.
We'll see. Okay. I'm always looking for new leads, like especially ones that are underrepresented in what, let's just say beetles or other insects or organisms in general.
To add to this project?
Yeah, once, because you see how I kind of find them to add, or anybody could do this to help add to this project. That you're doing it manually, and I'm first noticing one observation that has this American carrion beetle with a fungus, and I go, oh, that's possible, and I go searching through them. So I really need to find out about them first, and then I can, yeah.
Right. That was kind of what I was thinking, and hopefully my presentation is helpful to you. I think it will be in this agenda, but knowing the families that are associated with fungi would be really helpful. But also on the opposite end of that, a lot of polypores are associated with beetles because they're long-term life, right? They're perennial. And so they often have enough time to exist to support the whole life cycle of a beetle. There's also a lot of the tricholomas and the russulas host flies. So literally fruit flies. They're in the forest. So if you went by a family of fungi and looked for them, you might be able to see some weirdo that you know, some grandpa or whatever took a picture on iNaturalist with their kid of a polypore, and then you'll be able to see with your special eyes that there is somebody. Like, look at that tricholoma. Is there someone on there? Is there a rove beetle on there? Gosh, they're all tricholomas.
Yeah, sorry, did you see one?
At the top, it looked like there might have been a little rove beetle on one of them. It's hard to tell with like
Yeah, there's a lot of
but-
struts. One of the things
Yeah.
I sometimes do is I sort by popular because people love to see an animal on a. It's so popular. The filter is like based on likes. Like, let's see this. What's this?
Wow, this is
that
cool.
a moth?
Oh, look at that. Yes. I would say that's I don't know if you care. Do you care who it is? What the moth
I care
is?
who it is. Yeah.
Okay, it's probably a noctuid moth. In the larger group of litter moths are leaf litter moths. Well, that is neat. So wait, so that was an observation that you had not put in the project already?
No, I found this by taking your suggestion and just searching
Whoa.
tricholoma, and then I sorted by popular because people like
Oh,
it when they see a
oh yeah, double whammy,
animal
Pokemon
on
Snap,
it.
yeah.
Yeah, they love it. They love a double, so it's got at least one favorite.
Yeah, so the tricholomas, I've noticed a lot of things on them, and I think it's because they last a little bit longer and because they're big. So they're like, when was that taken? Oh, it was in
This is one from
September. Yeah, so like that's surprising. Oh, yeah, whole different part of the world.
from Russia. Yeah. It's not here.
Well, that's cool. That's a really fun like hobby, man. That sounds like a lot of fun. I will definitely. I have some stuff. My pictures on in my presentation are like mostly mine, and I've taken them from iNaturalist. So I did the first thing of like we're talking about beetles tonight, so I went Coleoptera, which is the order or beetles, and then I like was like, oh, there's one, and then I like learned about it and put it in there. And Coleoptera, man, I'm giving you all my slides already. But look at that, 11 million observations. So that's a huge. It's like, honestly, it's like the biggest group of animals on the planet. It is. So you'll have lots of time to look at the Coleoptera.
Look at that map, jeez.
Yeah, that's cool.
We're all just living in a beetle world.
Greenlandic beetles, that's neat.
Oh yeah, it's really fun to also. One of the things I like to do with fungi.
You could go by continent and just like wipe them out. Like that'd be really fun. Yeah. I wish I had time to do that. That's all I want to do all day.
And just go to some of these underlooked at regions too, and be like, who's there?
Yeah.
Same for fungi.
Oh, Mariah, you muted yourself. Someone muted you.
Oh yeah.
I think I can unmute you. Let me try that.
Why did she get muted?
You know, there's always some technical difficulty. That always happens.
Yeah.
Let me steal the screen then, Mariah. I'm gonna steal it from you while you figure yourself out.
Okay, I think I figured it.
Dang it. All right. Fine.
Oh, but you can steal the screen. You have a cool book.
All good. I don't have much to show. I should probably have my email when I do this. Let's try that again.
Okay, cool.
Cool. So I might recap some of the things you're already doing. You know, when we do these iNat things, we usually start with our project, our 2026 project, which Mariah already showed us a little bit here. I don't think we're going to see any bugs in this one. One thing that you were kind of getting on to the track here was like this species situation. So if you're in our project, you can see how many species we've seen. Let's go over that project that Mariah was showing us, this one called Organisms Associated with Fungi. And we can look at the species this way. And then Kristen, you were saying one of the key things is to look at like the families and get to know the families. So when I use iNat as like a resource for me to just learn stuff, it's really great to learn taxonomy because what you can do is now I'm looking at species, which is obviously very low, however you want to say it, in taxonomy, like very specific and species, and I can click on maybe the species, and I can see with this, where does this fit in the tree of life? So I'm gonna scroll down on this really cool spiky dude that I think Wes brought up in chat here. Both there is Cornatus, the forked fungus beetle, which you can definitely find in Wisconsin. We've seen it on a number of forays at like Scanoderma. And we can go down to taxonomy, and we can see where it fits, right? Obviously, it's in the kingdom animal, it's an insect, it's a beetle. We can go all the way down here and see the species, but we can go up to like family. So it's in the family of darkling beetles. And if I click here, now I can see all the other darkling beetles, and I can go like look at pictures of them, right? So this is an example of how you can use INAT to help you learn a little bit about taxonomy and other things. I'm gonna hand it back to you, Mariah.
Yeah, cool. There's a very loud helicopter out here. Sorry, I don't know if that is audible, but I was linking to some of the projects, and before I share my screen again, I'm gonna get the last link that I was trying to get to, which is lost. It was that one I was sharing earlier, the bioluminescent fungi one. Here, I got it from my own message earlier. Okay, this is the last project that I was going to share, and we can look at together. So I'm going to share my screen again and hopefully not mute myself again or however that happened. And this is a project that's similar to the one I was just showing, where it's curated. You have to add your own observation or the observation of somebody else that you find that meets the requirements of the project. And it's curated like that for the same reasons, that it's a very niche situation, a very specific situation. And it is arthropods. So anything in arthropoda, that includes all of the insects. It also includes things like spiders, among others. But any of the arthropods that you observe interacting with bioluminescent fungi, particularly while they're bioluminescing. So it could be that you're observing the fungus, but the fungus has a beetle, let's just say, on it, or it could be that you're observing the beetle and it's with the bioluminescent fungi. Either way, the goal is to help document these interactions and to serve as sort of an inspiration for some research that's now going on at UWL into this. And if you're like, oh, that's great, but there are no bioluminescent fungi near me, that is not true in Wisconsin, or almost, I would be willing to bet anywhere that there are fungi. Some of these are we're not going to see unless we travel, like this one, which is in Australia. But this is a gorgeous example of a perfect observation for this project, that the arthropod is visible, probably identifiable, at least to, I don't know, I'm going to make a guess of family. And then you can also see the fungus.
You could probably identify that to genus.
Oh, nice. You can, not me.
Someone can, yeah.
Yeah. Being eaten by a bug. Perfect. We love it when people use the notes fields.
I write goofy stuff in my notes all the time on INAT.
Well, look at Mariah. Someone looks like they put an ID down in the observation fields.
Yeah, they've put it to just grasshoppers, kits, and katydids. We could challenge ourselves to be like, could we get to genus on this?
It looks like it's a type of camel cricket. That's my guess. But it's also in Australia. So like everything there is like in a weird single, taxonomic genus.
This is where I love to use the comments. It's like the non-committal camel.
Right, yeah.
We could leave that there for everybody to consider. Also pop it a favorite so I get updates about it when people also comment. Going back to here, just highlighting one of the interact ones you could see in Wisconsin and surrounding areas. So here we have Omphalodus illudens are Eastern American jack-o'-lantern, not only called jack-o'-lantern because it's this gorgeous, jack-o'-lantern orange color, but it even glows too at night. And I've seen it in person a couple different times. Your eyes kind of have to adjust and it needs to be fresh. Also, the glowing seems to vary, maybe population to population, ? But this is also a great observation where we have what the fungus looks like in daylight, or probably just with a lamp on it. There's the setup. And you can see the glow. And I'm trying to see where we made out the bugs. I didn't do this one. So spiders. Let's see if we can spot the spider. Not getting it yet.
Take his word for it. It's there.
I'll believe in that. Maybe this was one that the observer, like Alan, added himself. Like he knew that there was a spider here and he wanted to document that. Or else I'm just not spotting it yet. But I will take his word for it. Oh, wait. There it is.
Wow. I would have never seen that.
That's crazy. Yeah, that sure is a spider. And it looks like, I don't know what this does for us taxonomically, but it looks fairly large and muscular as far as spiders go.
Muscle spiders.
Yeah, muscle spiders. But yeah, this would be even better if it had a picture that highlighted the spider. So part of also the project, the point of projects like these, like the one, like this one all about bioluminescent interactions, bioluminescent fungi and arthropods, or mine that is all the organisms just associating with fungi in general. Not only are these projects giving us stuff to be nerdy about and look at, they encourage people to document those interactions because, Alan takes great photography all over the world. And he takes a lot of, let's just say, these glowing mushrooms. So, like Alan, if you're out there, please take a picture of the bug so we can identify it. Got some, trying to think. I don't know. For our speaker, are there any beetles that stand out to you as interacting with glowing fungi? Or is this some new territory for you, perhaps?
What I've seen is pretty much the rove beetles on the Omphalodons, but I feel like Omphalodus, sorry, I combined their names.
That's fine.
The jack-o'-lantern fungus, I see rove beetles on them, but I think it's when they're pretty deteriorated already, something kind of peak versus deteriorating brings different kind of organisms to them.
That's a good point too. The bioluminescence does not always last either. This is one that I think is a beetle. Right? That looks cool.
That's a beetle for sure. Yeah.
Sure.
And it looks like a harmonia. Oh wait, actually that looks like a fungus beetle. It's hard because of the exposure, but
yeah.
And then also, what species of mushroom is that?
Something mycena in the subsection, and it's being observed in Costa Rica.
Ooh, lucky person. That is probably one of our triplex or tritoma beetles. I will give you guys my presentation as a PDF, so you have all these names.
Yes, thank you. This is one of the triplex ones. So there it is. It's all black.
Okay. Oh, did you type in triplex?
No, not yet. Oh, let's go check them out. Wow, I got it right on the spelling. I love these.
Yeah, there you go. I think it actually might be that first one there. You said it was all black, but I think that it just might be the lighting. That's
Yeah.
a pretty common beetle, but they're very small. So you're also relying on some super nerd like all of us to be like, there's something on something I care about.
Yeah. That's what you go to iNaturalist and Mushroom
Right.
Observer in these places for. See, this would be a premium one to add to my project because
Yeah, what's even going on there?
what even is that? It's a very shaggy.
Is
So
it
sometimes
is it a beefsteak? Is it like a messed up beefsteak?
I think it could be. I think
What
I don't
part of the
know.
world is this in, Marat? I didn't catch the location.
The location of this
North
observation is South
Carolina.
Carolina.
Huh. I would have thought like Pygnoporus, which is like I think more of a West Coast polypore, but out here we have like Orantoporus croceus. It looks a little too fuzzy for that, but possibly.
Yeah,
It's got like a coxcomb kind of thing going on where it could be like mutated and just look weird. Like if you look at that bottom left, that looks like a beefsteak.
this is where we can say was this fish.
You want to go for Fistulina?
Yeah, for Fistulina, is it Americana? Man, kind of crazy.
That's
Remember all
what
these
we're
things.
going with now, yeah.
I'm just gonna ask with the common name because it's a little late in the day for me to be spelling right now.
Also, do you know who that person is? They look like they're just like someone who saw something neat.
Yeah, quite possibly. You never know. You never know. But I will add, oh, somebody has, this is also a great one. I forgot about this project. Somebody on here. Somebody specific.
Oh, wow.
This person. Clearly.
T.C. Riley. I don't know who that is. I just want to say it like that.
Yeah, that's great. T.C. Riley. That's also how I'm going to say this person's name. So like is really, I'll just say it, obsessed with this pleasing fungus beetle family.
That's cool.
And yeah, that is cool. That's exactly what we like to see. And has this feeding preferences project. So every time I run into one of these, I try to also like put it into this too. So this is centered around this beetle, whereas mine was centered around you're interacting with a fungus. This is centered around the beetle. And yeah, just sitting here and going through and adding them to mine is one thing I've been doing. That's gorgeous. Just an aesthetic judgment there. So this is one of the ones I see a lot. Or that, no, sorry, this one actually, the Megalodacne Heroes one in Wisconsin. Oh, this is going to be fun to go through later. So we're about 2 minutes till 7, so I'm gonna quit and have perhaps Brennan introduce our speaker. But I really encourage people to get into these interactions. There's some of the really rich things that we can record as citizen scientists in the field in particular that then can provide like the basis for research, people having fun, gorgeous art. I encourage everyone to draw it too. So anyway, thanks so much for everybody showing up, and I'll turn over to Brennan to introduce our awesome speaker.
Yeah, if you don't mind, Kristen, I can give a shot at introducing you.
Yeah, sure, it's fine. Yeah.
You can go after I mess it all up. I only know you as KW, the hiking scientist from Instagram. I've been a follower there for quite a while, so I reached out to you to come do this. But I know you've done like mushroom circuit stuff before. I know you've been at like NAMA as a speaker. Obviously, you've got a lot of credit as just a general naturalist.
Thank you.
I think a lot of people here probably follow you on Instagram. I don't know if you do any other social media.
No, I don't. I'm old. I'm old, and Instagram's getting older. So someday I won't exist at all. Isn't that exciting?
Super exciting. Great, congrats. But your legacy will live on Instagram forever, probably somewhere, right?
Oh, thank you.
Cool. So KW, the hiking scientist, Dr. Kristen Wickert. I think I was following you before you were a doctor. been quite a long saga for you. You're like such a natural at the social media thing, which is probably why you have such a large following. But yeah, was there for your doctor thing. Saw you go through the whole thing with the Forest Service. Went through your Hot Kevin saga, which was kind of fun. You're like your own soap opera sometimes,
Wow.
but all good stuff, man. That's really all I have to say about you. Hopefully, I didn't embarrass you too much. Do you want to do like a better introduction for yourself?
Sure. You guys are funny. You're very, very smart, and like you've covered a lot of my slides already. So it'll be fun to go through these. But yeah, I do have like kind of an introductory slide, and usually it's because like I am getting older, so maybe I don't have to do this anymore. But like I've been a young female in science for a while, and so I always like have this like street cred slide to like show that I kind of know what I'm talking about. And this topic I have actually worked on, like I have publications about beetles that are associated with fungi that we're going to talk about tonight. So yeah, I've got a lot of pretty pictures and kind of a little bit more detail about why I am talking about the topic tonight and what my history has been associated with it. But you did great, and it's very funny. Like it's funny that Kevin's upstairs. I wonder if he heard Hot Kevin. But I can share my screen if we're ready for that. Do you think we're ready?
Yep, we're at 7:02, so let's get fungus beetle going.
All right, let's get cracking. So I'm going to move my screen around really quick just so I can like still see your guys' faces. And the way that my brain works is that it would help me if you guys saved your questions until the end, just because like I'll like lose track and it'll be like a train wreck. So we'll do that in a second. And then I'm on Zoom here and I can click share my screen and it should be the right one for that is in presenting mode in 3, 2, 1. You guys see a beautiful slide that's orange and black like Halloween?
Hell yeah.
Okay,
Really
great. So we'll try to go through this and I'm going to talk for about 45 minutes. I have 45 slides, so we'll see.
That's good. I have a quick question. Does the moratorium on questions till the end also count for the chat? Like, does that disrupt?
Oh, no, the chat. Go wild. Go buck wild in the chat. Thank you for asking.
Right. No problem.
So I really enjoy the fungus beetles because they're so, just like we were doing on iNaturalist, like they're just fun to look at and like see them interacting, and they during certain times of year, a lot of them are big and beautiful and we can see them. I also have kind of an affinity for them because they remind me of my high school. They are my high school colors. So this was my high school mascot, the Concrete Kids. It's literally a man made out of concrete, but the colors match the pleasing fungus beetle that we saw today. And hopefully our talk will be a little more interesting than this guy. But like I said, I often have this slide here to kind of show you, I know a little bit about what I'm talking about. There's always something more to learn every single day. But my name is Dr. Kristen Wickert. I've had a doctorate now for about 7 years. Geez, time flies, really scary. But I have a bachelor's in forest biology. So very, very broad beginnings. And then when I got my master's and my PhD, I really focused in on a field called plant pathology. And then even within that field, you can be a super specialist, which I am. I conferences for plant person that works on trees. Everyone else is working on soy and corn and things that have money associated with them more than trees. But I worked specifically with forest fungal pathogens that were fungi and insects. So I am both an entomologist and a mycologist as a plant pathologist. So a lot of these insects, and some of them will be beetles, they're exposed to our environment just like we are. So things attack them. They carry things to attack things so they can eat. And I used to study, and I still do, a lot of organisms and how they relate to everybody in the forest to cause disease or to be a healthy forest. I used to work for the Forest Service, and so I spent a lot of time traveling the United States. And I actually have been to Wisconsin to work. So I have been to La Crosse, and we worked on the American chestnut fungal pathogen, Cryphonectria parasitica. So a lot of the fungi I work with are not the beautiful mushrooms that are on iNaturalist. I work with fungi that grow on Petri plates like this, where they are just hyphae. They never make a beautiful mushroom, but they make very beautiful things in the microscope that you can see, like spores arranged in certain ways. And they do really cool stuff like eating spotted lanternfly, which you guys don't have yet, I don't think. And, interacting with our native millipedes that you guys don't have this either, but we have it here in West Virginia where I live. These feather millipedes, they are fungal farmers. So I study those. And then a lot of fungi that can be used to control bad guys in the forest, like hemlock woolly adelgid. I worked with a fungus that killed the adelgid, and then I work with a fungus right now that kills tree of heaven, which I know you guys have that. So lots of stuff going on in the forest. And I recently published a book about plants. So that was, honestly, I had another picture of me with the Forest Service there and it was making me sad. So I deleted it and I put that picture there. But I am now a professor of forestry and we'll see how long that lasts. But I'm glad to be here. And we're going to talk about two of those main subjects that I have specialties in, entomology. So we're going to talk about what is a beetle. And I know some of you know, but you never want to assume. There's always something new and fun to learn. And then what is a fungus? And you guys know that. So I made the slides really, really short on the fungus ones. But again, never assume. You might have someone here who's here for the first time tonight. So we'll cover how beetles interact with fungi and their symbiotic relationships. So that's mostly what I have focused on throughout my career. And we'll talk about there's like really dependent ones and not so dependent ones, and then ones that a relationship that happens that if one of the partners, fungus or beetle, is gone, they're dead. They can't do anything else. Really interesting. And then some relevant species for our part of the world. I noticed that when you guys advertised my talk, you had that cute little forked beetle there, that forked fungus beetle. We're going to talk about them because they are common in your state. So what is a beetle? It is in kingdom Animalia. So it is an animal. And like I said, I'm a professor now, and I ask every class, I say, what are insects? And some people do not know that they are animals, right? And I ask, what are they? And they don't know. So, but they are animals. And they are an arthropod that has jointed legs. And they are an insect that has 6 legs, right? So they have 2 pairs of 3 legs, 2 antennae, and then they have these 3 distinct body segments. So that is an insect, and that's how it's different than a mite or a spider, right? 8-legged spiders. And within that, we can go all the way down through the taxonomic orders of family, genus, species, right? So this is the elderberry beetle, very beautiful. That is in our state. That's my state of West Virginia's state beetle. Pretty cool. It's our college colors. But beetles represent the largest insect group with around 400,000 species known worldwide. And I always love to tell people the word known because we are very young in our understanding of the place that we live in, and we're finding new things every day. So this is not a typo. 1 in 4 animals known to science is a beetle. How fun is that? Not that old of an article. But like I was saying, if you want to look and identify all the coleoptera on a fungus, you're going to be looking at that website for a long time. But it is important, so go for it. It sounds like it'd be fun. But they are incredibly diverse, and they are distinguished from other insects by their forewings, which are called elytra. And they are hard, and they cover their abdomen. So you can kind of imagine this. Everybody's probably held a beetle in their life, right? You can like kind of click on their back. It's kind of hard. And they protect other very gentle or delicate wings underneath these that they use for flying. So they kind of hatch back up those wings, those hard elytra, and it exposes their membranous wings that allows them to fly. But because of those hard elytra, they can be in a ton of different environments because they're tough. They can like dig through ground. They can dig through logs because they don't have to worry about beating up their wings. And all beetles have chewing mouth parts, which you can imagine is going to be important for our conversation tonight. Insects are first distinguished from each other by their wings, and then they are distinguished by their mouth parts. And so all beetles have chewing mouth parts. You can see they kind of have these like adaptations that look like teeth on their mandibles, and they serve different functions, right? You can imagine getting bit by this guy is going to be a lot more severe than getting nibbled on by this dude if you were like a little tiny bug. That was their food. But beetles also go through complete metamorphosis, which is something that was kind of mentioned when we were talking about what orders of fungi to look in, or groups of fungi, I should say, because polyporaceae isn't really a thing anymore, kind of. But when you look at the polypores, they're a long-lasting organism in the forest, right? They're perennial. They keep growing every year. So you can have something that has a long life cycle, like a beetle, completely changing their body throughout their life cycle, reside in them. And so beetles will completely change as they go through their life cycle. And that was the Asian lady beetle, which you probably have in your garden, right? In the springtime, you have that little thing crawling around. But it starts out as an egg. So that's the first life cycle that it goes through. Then it becomes that creepy little dude, that little larva that does not have wings, and it crawls around and it eats aphids on your vegetables. And then it will become a pupa, which this is like probably the thing I get emails the most about from people saying, what is this? It's attached to the plant or to like the side of your house. It is a pupating lady beetle, and it will hang out in there for a while until it forms its full developed wings. There's those hard wings. We've all held a ladybug. And underneath are those delicate wings that it uses for flying. So as the larva, babies, they may have different sets of needs than they do as adults. And that's what's important if you guys want to try to find insects associated with fungi. There might be a time period where they need to eat the fungus. It matches up with their phenology. Or they might change and they become predators of something on a fungus, right? But all beetles, during all of those life stages, well, the nymph and the adult have those chewing mouth parts, and they eat. So they can be, often they're predators of other things. And just because you guys are nerds, I wanted to include this. And this is actually my photo here too, where on the little backside of this Asian lady beetle, there is something growing right there. And the something growing is actually a fungus. So Hesperomyces virescens is a sexually transmitted disease of our lady beetles. And you can see this like usually like July or something like that. They will have kind of this sexual transmitted disease on their backside where another beetle would come, try to mate to make more baby eggs, and it would get infected. Pretty crazy, right? So we are not the only species that gets problems from a late night out. But there is so much diversity in the order Coleoptera that beetles are in. Like this is like, it was hard to find a picture that showed what I was looking for, and of just like all the different like sizes, like that thing, that's as big as your hand. Like it's huge. And then we have some really, you know, look at this guy. They all have different body shapes and sizes for the different like jobs that they do in our ecosystem. And there's this really interesting like quote from JBS Haldane, who is an entomologist that just, it's always cited in the field of entomology that the creator, if there was one, he was actually an atheist. But he was like, man, if there is a God, they have an inordinate fondness for beetles because they just kept finding more and more beetles. And when, like, you know, that was back in like, you know, black and white photo days. But today we have the ability, just like you guys with fungi, I'm sure, we have the ability to look at their DNA and try and figure out where they belong and like how closely related they are and like what jobs they've had. And it's so interesting in this phylogenetic tree, which we'll just observe because it's way too intense, right? This could be a scientific paper and report for a college student. All of the beetles are grouped into different families here. And this really big group, Polyphaga. Poly means many. So phaga means like consuming. So these are like all these different weird beetles that get lumped together that eat diverse things from each other. And we have our little fungus beetles. They're younger, and they have records of being present in the Jurassic period, the Cretaceous, and the Paleogene, and today, obviously. But Jurassic is like, we all know the movie Jurassic Park. The tropical, like carboniferous forests happening are when fungi started to exist. And then also these fungus beetles started to exist. How cool is that? So we have a lot of like different things going on in just one group of animals, all meeting different needs. But what we can talk about without looking at a scientific paper is what are these beetles even doing in our fields and forests of Wisconsin? Well, they're all eating with those chewing mouth parts. And there are different kinds of groups, like you saw on that phylogenetic chart that said polyphagous, like they are all eating different things. So a lot of our beetles eat plant foliage. They eat the roots, they eat the flowers, pollen, nectar, seeds. Pretty much how they look reflects the diversity of what they eat and what they do. And if you have like a native plant garden or you have beautiful wildflowers on like your driveway, you probably will see this beetle that's always sticking its face into a flower eating the pollen. If you're a gardener and you have rhubarb that suddenly died, it's probably because this guy ate your rhubarb roots, this rhubarb weevil, which is a native beetle to us, even though rhubarb isn't. Then you might experience a really scary dude that's like bigger than an inch, this Prionus lacticolus. It is the broad-necked root borer that eats our forest tree roots. And then we even have little tiny beetles that live inside of a seed box seed capsule that the only thing they can eat is the seed box seeds. So we can have really hyper-specific things in our backyard in the form of beetles. They eat wood living and dead, where often fungi are too, or they're like a precursor to fungi being able to get into trees. But if you have any dead trees, and I actually took this picture in Wisconsin when we were visiting looking at a pathogen we'll talk about, but this oak timberworm is a key destroyer of wood in that it has enzymes that allow it to digest lignin and cellulose inside of its body. So beetles can be eating hard things like wood, like the horrendous emerald ash borer and the oak timberworm, and they can also eat other insects. So we have this fastest animal proportionate to their size. The tiger beetle is a very aggressive hunter, and it catches other animals in its mouth with those sharp mandibles, those chewing mouth parts. We have these carabids that are called the snail hunters or the worm hunters, and they come out at night and they eat unexpected other animals. And they even use these beetles, their ability to be predators. So these little dots here are hemlock woolly adelgid. They kill our eastern hemlock trees and are a very serious forest problem. But we deploy little beetles that are specialized in eating them to help restore our forest's health. And this guy, I'm so glad you mentioned them. There are beetles who specifically eat only dead things, but sometimes they visit fungi because the fungi are trying to smell like dead things. So the carrion beetles that you guys were seeing on the stinkhorn are likely confused, and they might actually not even be eating the mushroom. I don't know, but it seems that the mushroom or the fungus is trying to trick this obligate dead animal eater into thinking that there's an easy meal there. So if you've ever smelled a stinkhorn, you know how you want to like gag and like get as far away from it as you can. That is an intoxicating smell for our Native American carrion beetle and other species like the hairy rove beetle. So these guys eat dead materials, but you might see them confused on a fungus, thinking it's a snack. And we even have beetles that will eat poop, and that's all they eat. They only specifically eat animal poop, like our dung beetles. And I know you've probably seen dung beetles on, like, you know, I don't know, National Geographic, and it's like always in the tropics. You have them probably in your backyard. This picture was taken in my backyard. These are these little Onthophagus hecate, and they go out into cow fields and they make dung beetles that they take down and into the soil, and they lay an egg in it, and their life cycle is solely dependent on poop. But finally, this slide that we care about, because I bet some of you were like, I'm not going to listen to this lady talk about beetles forever. We're finally at the point where we're talking about fungi, and there are beetles who only eat fungi. So there's the beetles who eat only poop, only dead animals, only other animals, and plants. And then this really interesting group of beetles that only eat fungi. And so as an example, and we'll have more details on a bigger slide, but this forked fungus beetle, Bolithotheris cornutus, is eating a mushroom that it will also lay its eggs in. So it is completely adapted to living its life right here on this tree, and it's not going to go anywhere else. It has no reason to go anywhere else because its food and its mating source material is all there. Same goes for the red-winged tritoma. These guys fly a little bit farther because they're often found on forest floor mushrooms, and they have a really quick life cycle. You can see these guys are getting it on. They're mating because they have to lay eggs in that mushroom. Their babies have to eat that mushroom. And then, you know, how long does a hexagonal polypore last? A couple of weeks, maybe, before it gets all dried up, right? So there's a lot of big rush happening in these little beetles' lives that depend on fungi, which depend on moisture in our forests. So this is the slide. You never know, this might be someone's first mushroom club meeting, but we always want to talk about precursors to what fungi are. And same thing, fungi are not plants. We all know this now, hopefully at this point, but they were lumped in plants for a long time. Like some of us have field guides that still say like spore producing plants, and it's pictures of ferns and a picture of a mushroom. No. So 1960, it was the era of when fungi, the kingdom, was made separately than plants, based mostly off of the fact that fungi are made out of chitin, not cellulose. They reproduce by spores, not seeds. They grow as hyphae, not roots. And they also are not autotrophs. They don't utilize sunlight to make their own food like plants do. They don't have chloroplasts and chlorophyll. They need to eat something else. They're very similar to us in that they dissolve their food with enzymes. So when we eat food, we have enzymes in our body with the help of some bacteria that allow us to break down the food into sugar. Fungi release enzymes into their food to break it down into simple things that they can absorb and use for energy. So just as a quick preview, I know you guys all know this stuff, but they're more closely related to us as animals than they are to plants, which they have been lumped in for centuries, right? So they grow as hyphae through their substrate. Here they have their enzymes that they're secreting out of their hyphae into their substrate, which dissolves them, and then they pick those sugars back up and use it to grow. And sometimes the hyphae grow into a mushroom that we see. But remember, I worked on fungi that only existed as this, right? They only existed as hyphae, and they make spores. They never make a big, beautiful mushroom. And I think that there's a lot more fungi that exist in the world like that than there are with the big, beautiful mushrooms, because they're living inside of many different things where if they're inside of their food and they have no reason to make a mushroom, why waste the energy? So they live inside their substrate, and they are just kind of this cryptic thing in our forests still. But when we combine those two things that we've given an introduction to, the beetles and the fungi, there's a lot of different ways that they interact. So often what you see when you're looking for iNaturalist stuff is that beetles will be using fungi as a hunting ground for other insects that are in or on the mushroom. So a really common one that I find in the springtime, right, because this is a morel, is this Photurus firefly larva, which is a beetle. Kind of weird, right? You don't think of fireflies having a distinction, but fireflies are beetles, and they have enzymes in their butt that make them glow or whatever, but they are larvae that have chewing mouth parts. So they are hunters. And they could eat the mushroom directly, but they are just really voracious, aggressive predators. And this is another genus of firefly, which is very different than this guy over here, even though they look similar. And it is on the underside of a maitake. And if you look above them, you can see that little fleck that looks like dirt. That is actually a little tiny fungus gnat. So that is like what you get if you like go on vacation and you forget food on your counter. You're gonna get fruit flies and you're also gonna get fungus gnats. So this little fungus gnat, that's an adult with wings, this aggressive, crazy dinosaur looking thing is probably trying to eat that and to eat its babies. So the fungus gnat, like its name suggests, needs fungi and it lays its eggs in here, super fast metamorphosis. The larvae eat the fungus, they pupate inside, and that's like when you cut apart a mushroom, you see all of the trails inside. Like you've ever cut up a mushroom and you see these little crazy trails? Those are because of fungus gnats, often. Sometimes it's other things, but it's often fungus gnats that look just like that when they're adults, but you never see them as adults. You're seeing them as their larvae inside of the mushroom. So our beetles are very hungry and trying to eat those guys who are protected inside of the mushroom, but as they start to come out as the adult like this, they are a free snack. Another example of that is this group of beetles called the staphylinid rove beetles. And they have these funky little butts. They run around. They don't fly very much. They have very small elytra. That box right there is their wings, very, very small. But they run. They're very good at running. And they have these little butts that they stick up in the air, and you can see them looking in all of the little crevices in the pore surface of this mushroom for other animals. They're not eating the mushroom, and they're often on mushrooms that are like slightly decaying, and it's because of the time period of the fungus gnats had enough time to eat it, they're starting to pupate, and they're coming out. And so these little guys are having a fiesta. They're having a party, eating all of those fungus gnats that are inside of all these crevices. They may also be eating springtails, which are another type of arthropod that are very small that live on mushrooms sometimes and do eat the mushroom directly. We do have beetles that directly eat and rear their young, and they are obligate feeders of a specific mushroom. So this is the four-spotted beetle, four-spotted fungus beetle. 1, 2, 3, 4, 4 spots on the back of their neck, right, one of their body segments. And they can have kind of a weird Rorschach pattern on their elytra, those hard wings, but they'll be a little bit different from individuals, but they'll always have those 4 spots. So this is the four-spotted fungus beetle. I don't know what it's eating, maybe an urpex, maybe, right? Some kind of crust fungus, but you can actually see their chew marks, right? See like the dimension and the, layering that they've eaten down. And you can see that there's actually like some depth there. So these adult beetles, oh, and look, you can steal that too there. I stole this. It's someone's iNaturals observation. So you can see that they've been eating it directly, but once they get their fill and they get enough energy, they're going to mate. They're going to have a good time, eating, drinking, and hanging out in the fungus, and they will lay their eggs in there. And their babies, remember, it's a larva. They have that complete metamorphosis. So this might look like a maggot to you. It might look like a fly maggot, but it's actually a baby beetle that doesn't have his hard wings yet because he hasn't gone through complete metamorphosis yet. And if we look at these two pictures, look at how fresh and white that urpex is, if that's what that is. It's super fresh. The adults are eating it. And here you can see it's got that old, like yellowy bacterial, like it's crusty. It's old. It's on its way out. but the babies have developed enough in there. And so you'll actually see the babies later in the season than the adults because of their like need to be an egg and a larva and then an adult. And so sometimes the adults will overwinter. It's different for every species. All right, so there is actually a family like of fungus beetles, right? It's a big group of things. And when you Google, and you'll notice I'm trying to use Google Scholar, so I'm actually trying to find real stuff, not AI garbage nowadays. But if you use Google Scholar, and even if you so that's like a criteria of like higher quality stuff already. But if you type in role of fungus beetles, like trying to learn like about their ecology or whatever, there is not a lot of stuff that comes up because they're kind of not doing anything that bothers us. And as humans, we often care about species that cause problems for us. And so you'll get things that talk about the ambrosia beetles, which we'll get into, that cause a lot of problems, and therefore they get funding for research, right? Because if we have a beetle and a fungus that are killing a tree that we care about, we're going to investigate it a lot, versus these beautiful little fungus beetles that are just kind of these cute little things in the forest. There's not a lot of research. So if I type it in, fungus beetles with an actual like scientific genus there or family, there's information, but it doesn't really talk a lot about what they do. There's not that many results. So it's hard to find information about the good guys, which is what you myco lovers see on the forest. But when you try to look up role of fungus beetles, you're going to get 111,000 results that are about bark beetles that cause problems on timber products. So it's hard to find information about these guys on the internet. And I actually used a real book. So I went and found a book at a museum and I learned about the different types of fungus beetles, which covers many different families. So you will see the families, and this will be really helpful for you when you're digging through iNaturalist, this list of families here. Family Erotilidae are the pleasing fungus beetles. And we are going to talk about that group. But there's a lot of other stuff here, right? The silken fungus beetles, it's referring to how they like comb the fungus. Cryptic slime mold beetles, which are not fungi, right? We know that slime molds are not fungi, but they often get lumped in together. Handsome fungus beetles. Why is that different than pleasing? I don't know. And then we have the minute bark beetles and minute tree fungus beetles, which I want to talk about today. And then we have the darkling beetles with our big one example. This is a huge family of beetles, which is mostly like snail hunters. They mostly eat like snails and other animals, but we have one fungus beetle. It's very obvious in your state. And then the Curculonidae, the bark and ambrosia beetles. So there's a lot of information if you go to a museum and have an entomologist help you, but there are a lot of different families that are grouped into interacting with fungi as the fungus beetles. But they often have a similarity with their coloring, and we're not really sure why, but scientists, this is actually, Dr. Powell is in North Carolina, near Raleigh, and they are looking at the diversity of the colors of fungus beetles, and they're usually always orange and black. Okay, so let's move into our first family within the fungus beetles. The pleasing fungus beetles feed on the fruiting bodies of fungi, and they don't go after the hyphae that's growing through the substrate. They're looking at the fruiting bodies. And a wide variety of fungi serve as the host for this family, so they're kind of generalists. They're not super specific. They're not locked into only being able to eat this one mushroom in order to have their babies in it and to live their whole life cycle. There are a lot of things that they can be found eating. But each fungus beetle kind of has like a preferred group within that. So it might go after the Ganoderma, right? But it's not always constricted to going after Ganoderma apellinatum or Ganoderma sugae. It goes after the Ganoderma. But the pleasing fungus beetles play a role in the ecosystem by aiding in the decomposition of wood and the breakdown of fungal material. So fungi, we know, break down trees and wood. We can thank them for being able to walk in our forest without tons of logs and branches. But we need something then to break down the fungus. And here we have these beetles that are specifically made for eating mushrooms. And when I looked up, see, I like iNaturalist too. When I looked up this family of pleasing fungus beetles, I found that there are 17 common species in Wisconsin with a wide distribution. iNaturalist is always interesting to consider that, you know, you have a lot of people that live down here, Madison, like people, you know, in college, right, using iNaturalist. And it's not saying that there are not beetles associated with fungi up here in the national forests. It's just kind of hard to get up there. And like, you know, if someone's camping, they probably aren't looking for beetles. They're like, you know, kayaking and all hiking and stuff like that. So you have a broad distribution of these fungus-loving beetles throughout your entire state. And the ones that you have probably seen are the Megalodacne hirose, that four-spotted fungus beetle. And then we also have this breast-plated pleasing fungus beetle, that triplax. Some of these other beetles, I'm not really sure, like they're like put into the family Erotilidae. However, there's a lot of reclassification going on in beetles, and a lot of times these beetles were orange and black, and they put them in the family pleasing fungus beetles. Because this beetle, which has the most observations in this family, besides your big old pleasing fungus beetle here, they are not associated with fungi. They are, and you can even see it's on a plant here, they live and they bore inside of the stems of like asters. So there's a lot of reclassification that should happen, but remember there's 400,000 beetles. So it's a big job to try to like figure out where all of these things truly belong, with a bunch of different people who have different opinions and always trying to edit where a family belongs or genus. But this species, I've said its scientific name, but its common name is the pleasing fungus beetle. It is the pleasing fungus beetle in the family of pleasing fungus beetles. Here you go, you can see going back through its classification. So here's its species, genus, and then its family. So this is the pleasing fungus beetle. And it is most frequently found feeding on Ganoderma tsugae. So if you really want to find this beetle, you have to go somewhere where there is eastern hemlock growing, because the fungus Ganoderma tsugae only grows on eastern hemlock, which has the Latin name Tsuga canadensis. So there are few scattered records from other polypores, which your group might be able to help going through iNaturalist to understand what other things they can eat. But really, I see this beetle in April to May where I live, because you guys are a little bit more north in latitude. It might be a little bit later, it might be June, but really you have to think about where the adults come out from under the bark, they hibernate over the winter, and they will lay their eggs in here, and this mushroom will probably still be growing as their babies are in there eating. So they have to like get all of their life cycle within this really short annual polypore. So this is one of those polypores that does not get bigger every year. It shows up once like in the, you know, early summer, and then it deteriorates. So this beetle is really linked with the phenology of Ganoderma tsugae and the specific habitats too. And we actually have two species within this genus, and they're very, very specific. And I am a trained entomologist, and so this is actually from the dichotomous key of the two species in this genus. And I mean, if you just look at that, you're like, where? Like, what is the difference? And it's one of those species differentiations where it requires you to actually like measure stuff, right? 9.8, 15.5 millimeters long versus 14 to 22 millimeters long. And the shape of their pronotum, which is this part, this like shield on their neck, the shape is curved versus square. Okay, but they, you know, they have their own thing, right? So a species means that they can't, like, if it mates with something else, they're not going to produce viable offspring. So there's something else going on in there versus how they look that allows them to be their own distinct species. But these are the two species you'll find, and in Wisconsin, it looks like you will experience the Megalodactyl heros more commonly than the fasciata species, which is a little bit smaller and has a weirdly shaped pronotum. The next species that you'll probably find on a hike is the four-spotted fungus beetle. That's that one that we talked about, 1, 2, 3, 4, that lays its eggs on the crusty kind of species. So it feeds on polypores, but also on some crust, like fungi of decaying wood. And they are gregarious. I don't know if you remember spelling bees in elementary school, but gregarious means like, you know, they are with other organisms, they're talkative, right? And they're social. So you will often find more than one of these in a spot. So they will hang out as a community. And so the adults will be seen together feeding, and then their babies will be together feeding as well, all on the same fungus, right? Because these guys don't have wings yet. They can't fly away, and they probably don't even really have well-developed legs. But I have seen them growing or feeding on succulent fungi in April to May. So earlier in the season, because they need to lay eggs and their babies need to live out their whole life cycle on the food before it goes away. So the last example in that family of the pleasing fungus beetles is someone who doesn't actually eat polypores. It's more commonly found on our bigger, more fleshy mushrooms. And the genus Triplax has a lot of species in it, and they're very confusing to identify, but mostly what they have is that orange head and pronotum and then a black body. And again, these are my photos too, so I'll figure out which one it is and I can add it to your project, but they are feeding directly on the fungus. And they're pretty tiny. So I remember using like a magnifying clip on my cell phone to take these pictures. So these guys are really small, mostly like 2 millimeters long, but they are a big fungus beetle because most of the beetles are much, much smaller. Their family's common name is the brightly colored fungus beetles, referring to that bright orange head and pronotum. But you'll often see them on the soft bracket fungi like Pleurotus, not the hard polyporous or polypore mushrooms that are perennial. So now we're going into a different family, the Tibronidae, the darkling beetles, which only really has one big example, which is the forked fungus beetle. And it feeds directly on the mushroom that it finds, and the males will actually battle on there. So they use that horn to battle. They're like rhinoceroses. And they will throw each other off, and they will protect the mushroom, which attracts females to say, oh, you are so strong. Let me lay my eggs on here and you can protect them. And there was a graduate student that actually wrote a paper all about measuring the grip strength of these beetles by tying a string on them. So look at that. You can do anything. So we're getting into the set, like there's a few slides left, but they are the most exciting for me. I love these beetles. They are very, very tiny. So less than 2 millimeters. They're super small. You have to, like I took this picture through a microscope. So the family Ceidae are the shell fungus beetles, or the minute, not minute, minute tree fungus beetles. And they are less than 1 millimeter sometimes. They're very minute. And their body is short and cylindrical, often convex, and sometimes with a smooth coat of fine short hairs, which they collect spores on. And they are always found within or on polypore fungi. They do have wings, but they often don't fly around. They live their whole life on perennial polypores, and or sometimes annual, right? There's an annual one, but mostly they're in perennial polypores. And they're very cryptic and understudied because they're so tiny. Just how tiny? They're very tiny. You have to use a microscope. So this is a dissecting scope, and I'm probably magnified about 40 times looking at these guys here. I inherited a box of old artist conchs. Someone thought I could use them to teach with, and they were completely deteriorated. And in the dust, in the bottom of the box, were all of these little beetles that somebody picked off the artist conch, put it in a box, forgot about it, sealed it, and the beetles continued their life cycle, and then they came out. And they had nowhere to go, so they died in the box. But there were hundreds of them, suggesting that our conchs are likely filled with thousands of beetle eggs like this that you see and you take pictures of all the time. You're actually taking a photo of, theoretically thousands of organisms. Very cool, because they're so tiny, occupying that space in that mushroom. So this species I grew up calling Phelinus robiniae, but you guys might know that it has had a name change, like all things, and it is now called Fulvifomes robiniae. The common name is the same, the cracked cap polypore, which is a perennial polypore, that is hard and woody that grows on black locust. And inside of this mushroom is an entire system of beetles that leave these little tiny holes. It is called the Malacosis brevicalis, and when it is a larva, it eats and makes these little tunnels, which I took this photo with a microscope. You can see the rim, the eyepiece. The larva lives inside of this mushroom and eats it. Those are its little poop pellets, and it completes its entire life inside of this mushroom. And so this map was showing where you have Fulvifomes robiniae, the cracked cap polypore, and I'm going to show you this beetle here. This is the larva eating the mushroom. Those are its little poop pellets, lives its whole life in there. It's got some poop on its head, gross. And that's what it pupates, and then it turns into that. And this is a ruler. You can see that it is just over the size of a millimeter. Very cool. Again, photo was taken through a microscope. There he is saying hello. Very cute. But this is the number of observations for that beetle. I have gone to so many places and looked on the underside of a cracked black-capped polypore and seen all of those holes, which are caused by the beetle, but there are no observations of this beetle. Half of those observations are mine. And a lot of them, they never get confirmed or research grade because nobody knows anything about this beetle that lives its whole life in a mushroom. And we have another example of you could find a new species. I know you guys have this mushroom up in your forest, the Cryptoporus vulvatus. It grows on pines and it has kind of that veiled pore surface there. Well, there are beetles that are associated with it that no one knows anything about. And this is one I found, and no one was able to identify that beetle. So they guessed maybe it was in that Ceidae family, the minute tree polypore beetles, and maybe it's in this genus. But these guys require an expert to identify because they're so small. And I did some digging, and I found that there were some nerds that were in Korea that wanted to know the answer that I wanted to know. And they found that there were several species of beetles that were associated with the same fungus, Cryptoporus vulvatus. And they identified these beetles. And we do not have this beetle, but we do have this beetle. So this beetle could be associated with Cryptoporus vulvatus, just no one has seen it. Same with this one, not that one, and then this one. How crazy is that? These guys could be living in our forest associated with a fungus that we know nothing about. And the final family I want to talk about is the Curculonidae family, which are the bark and ambrosia beetles. And these beetles are so reliant on fungi that they could have you could talk about them for an hour, but we're only going to talk about them for 2 minutes. They have distinct parts of their bodies that they carry fungus in them. They're called mycangia, and they're denoted by these yellow little spots, these little drawings here. Oh, that question mark, we don't know about that one. But all of these weird body parts carry fungus. And symbiosis that's so serious and brain, that's the beetle brain, and these big things over here, these purple things, are the eyes. So those little eyes there. This with an M is called a mycangia. So this beetle actually has cheeks like chipmunks that are filled with fungal spores. And when we look at it really, really close, we can actually see the distinct hyphae in there. So the fungus is adapted to living in those cheek pockets, and the beetle spits it out when it goes into a tree, and the fungus eats the tree, and the beetle eats the fungus. And so this is a species that has its pockets, chipmunk cheeks on its back. There we go, those little spots right there. And there it is through a microscope. So those little pockets have yeast-like fungi in there that it rubs its back on the tree it drills into, infects the tree, and then it eats that fungus. And this is just some work that my lab did a couple years ago now, but we found a new species of fungi that had never been described before, and it was literally in the ditch out behind our office at the parking lot. So you guys can find new stuff. And this is what it looks like. So I said that the fungus comes out and eats the tree. Well, it coats the walls of the beetle's tunnels with a fungus material that the beetle can eat, because beetles can't eat directly wood unless they have the enzymes for it, but the fungus can. And so the beetle eats the fungus and gets its food that way. So the fungus in the trade gets transported to a new location where it gets new food. Pretty crazy. So sometimes we can use that to our advantage, and that's the research I do now. I utilize a fungus that kills tree of heaven. And there is a beetle that has those spots on its back that transport this fungus around and lead to more disease spread, helping us not have to use pesticides on a very invasive tree species in our forest. And so this is the last two slides. This is the honorable mention of fungus beetles. Technically, they're not really fungus beetles. They're more so attracted to like bacterial oozing and sap. Like when a tree is really stressed out, these sap beetles in the family Nitidulidae, that's fun to say, but they are attracted to like oozing wounds in trees. And they literally come out of those holes covered in slop and slime and spores, and they don't care. So this is something where the beetle doesn't care about the fungus at all. It's just existing, but it has like a mess all over its body, and it carries sometimes a disease which happens where you guys live. So oak wilt is a fungal disease that relies on those sap beetles coming to stressed out, stinky trees and getting covered in spores to go and crawl on another tree and infect it, so that way it can spread and move and eat another tree. So you have oak wilt throughout your entire state. I've actually been to this part of the Midwest to see it, and it's probably something you guys are all familiar with, right? You see these dead oak trees that die all of a sudden. It is because of a fungus that is carried by a beetle that infects trees. So there you go. I know that was a lot, but I mean, there's more. There's always more. And I just want to say thank you for paying attention, and get outside and go find something. It seems like your club is really active and looking at things that are often ignored, but that's how you find something new. So thank you. Thank you for the clapping emoji. I like that, Jessica. Thank you. There is some time, I think, for questions if someone wants to moderate that.
I usually moderate it.
I'll leave this up in case we need to blast back to
yeah, and we can also look at the structures there on that.
Oh yeah,
which is cool. Cool to see them up close. Oh my god! Thank you so much, Kristen. First of all, from all of us for speaking to us. We did have at least one question in the chat. People can write their questions in the chat if you want me to read it, or if you want to unmute yourself. I'm gonna do like pauses so that people can do that. So. Amber had asked a question in the chat, and it was, are beetles more active during the day, or are some nocturnal?
So that's super dependent on what species they are. The snail eaters, like those carabid beetles, oh, they are out at night. They are hunters. They are going for the snails that are cruising around that don't want to be in the sunlight because they'll roast. So there are a lot of nocturnal beetles, and there are also warm daylight beetles. A lot of these, like ambrosia beetles, will fly around in the daytime because they're cold-blooded, right? So if they're flying, they want to be heated by the sun so they have more energy. And it's just easier for them to move around. But I would say, you know, from what I know, 70% are active in the day, 30% are active at night where we live.
That's really cool. Also makes me think about how to bait beetles out of pollen at night or something. Like, do they come to a moth light?
Some of them do, yes. So I put out moth lights a lot, and some beetles come to it, but not a lot. Mostly moths.
Cool.
Looks like Andrew has his hand up.
Oh yeah, go ahead.
I don't know how to find this digital hand, so I'm using my actual one. I have a question. I have a very cool observation from Governor Thompson Park of rove beetles, which people believe is Oxyporus vittatus, I think. And it's eaten all over the red scabier mushrooms. It's got holes poked in it. Mariah can quickly find it if she can find Governor Thompson Park on iNet. There's not that many observations in there. It's named after Tommy Thompson. And I was curious whether it's actually eaten a mushroom, or is it eaten something inside the mushroom? It felt very comfortable in those red scabier caps. Very neat fungus. I love it very much, but I don't know what the hell it was doing in there. I'm not very knowledgeable in that sub group of rove beetles, so I don't know.
So I would guess, and of course I could be wrong, I would guess that it is ripping apart the mushroom to eat the other animals in there. When I was reading the actual real book about fungus, like any order or family of beetle that was associated with fungi, had any note about it, I would read it, and it said that the rove beetles are often on fungi, but really they're ripping it apart to find animals, like they're predators.
Predators. Okay.
But you could, I mean, if you have time and an obsession, you could sit there and stare. You could see what they do.
I found the observation. I put it in the chat.
Ooh, yay, thank you.
Yeah, maybe throw up on the screen if you don't mind.
Yes, you are relying on me to be smart enough to do that, but I can try.
We
Okay,
could always figure that out too.
It's okay. Wait, I know where it is. Nope, I don't. It won't come up. Sorry, I don't know where it is. I can stop sharing though. How about that?
Sure, I could try sharing my screen and then looking at it.
Okay. All right, cool.
So real quick, we will see. We'll all get to see it. Oh, as soon as my computer decides to let me.
As soon as I can stop sharing, it's really, this is embarrassing. I'm on Zoom all the time.
It's not you. It's okay. So we have Andrew's observation on a red lexinum, one of those like foxy boletes. Oh, it's such a pretty little rove beetle. I think, is this a rove beetle? Am I wrong?
Yeah, it's a subgroup of rove beetles.
Okay.
There's a lot of rove beetles, but they're very characteristic. So they have a lot of fans. I think I finally stopped sharing. It was like hidden. It was like, I don't know where it went. It was just crazy. Oh, but look at that. Whoa, that is crazy. Oh, wow. That's really cool.
Yeah, and then it looks even cooler on top of the mushroom. Mariah, show the top of the lexinum. Looks like a pepper thing, whatever.
Cool.
Pepper, salt.
Like a pepper grinder. Yeah, or like a chocolate chip cookie.
Yeah.
I think that looks like, it does look like a cookie. That's funny. I think it looks like it was just ripping it apart to get what was inside of it. Because you're not seeing like frass mess all over. You're seeing like it's just pulled to the side. Like it's not, I don't think it's eating it. I think it looks like it's just like digging it apart to get to the thing inside of it. Man, that's cool. That is really cool. I'm gonna send that to someone.
Yeah. Maybe if next time, like if we ripped it apart, we might be able to see the smaller
tunnels and who's in there. Yeah.
Evidence. Oh, and this
Wow,
brings me
that's so
actually
cool.
to a question I saw DJ ask in the chat, which was when there's those like tracks that are left by an eating like fungus, gnat, larva, or whatever,
Yeah.
are those called galleries like they are in
Yes.
plants, even if they're into the fungus?
Yes. Yep. We call them galleries.
Cool. That could help someone when they're doing their own little Google Scholar research for sure. Well, I'll quit sharing here, and if you need to go back to your PowerPoint.
Yeah.
All right, so we asked DJ's question, and then Mycological Society at UW-Madison has asked, do you have any advice on the best ways / places to find fungus beetles besides them being on a mushroom?
Nope. That is it.
Find the
And
mushroom.
they're out during the day, so you see them on hikes. I wish there was a more exciting thing.
It's an exciting thing. Beth is suggesting under bark, perhaps. Like, would we find?
Right. So the pleasing fungus beetle estivates underneath bark. So, but I mean, I do that sometimes, but I've actually like morally stopped because you're destroying its home. Right. Like, and I get it because we all grow up like rolling logs and looking for things, and that's how you fall in love with stuff. But then it's like you start to actually think about it and you're like, oh, I just like ripped this thing's home apart and then I'm gonna leave literally a minute later. And I've just ruined its life. Like, so I've started being a little bit more considerate of that.
Yeah, I definitely get that. I feel like it's a strong balance, and it's one of those things where you also think about like the local abundance of this, or like which requires that ecosystem knowledge to start with. You know, how
Right.
much do you want to risk? Which
Yeah.
that was something I was just gonna bring up, that we've tried to discuss perennial polypores as habitat. And it's something that we don't rip down unless we have a strong purpose for it, because it's habitat. And also you could hurt the tree if it's still on a living tree. But you know, like those beautiful artist conchs that we love to do the art on, I do feel like it's still a balance. Like that's a lovely folk art, and also they're very locally abundant as
Yeah.
the species goes, those Ganoderma applonatums. But they are habitat, and you have to know that there's beetle eggs in there for 2 reasons, so that you know that it's habitat and you don't tear them all down for no
Right.
good reason. And also so that if you do take one and you draw on it, you heat kill the beetle.
Hundreds of them.
They will
Yeah.
come out into your apartment. I've discovered this too.
Yeah. Yeah. So I used to be the president of the West Virginia Mushroom Club, and I still go to the forays. I'm speaking at it this year. It's in August if anyone wants to go. It's a beautiful time. But we have about 250 people. And when we first started doing the forays, you know, everybody goes finds mushrooms and bring them back. And we have like permits to do all this. But we realized that there was like a table of like the first year there was like 50 artist conchs, and then the next year there were 5. And then so now we have a rule that like someone, like one of us, the officers, brings an artist conch or something from like home or somewhere else, and we don't decimate those perennial conchs. And we usually we make kind of a big deal about it in the beginning, but that's another perfect example of like you don't learn this stuff until you do it, and then you see, oh, what have I done? And I was just, I was loving nature to death. That's what we were doing.
Yeah, and it's definitely a balance. Or like, will I always not take a bracket fungus?
Right.
No, I will sometimes, but it's like I have
Yeah.
a purpose, whether it's scientific or artistic or culinary, some purpose, and I'm going to follow through. That's like a classic forager's rule. You have too much if you're not able to follow through on what you've got.
Right.
But
yeah, but they're just out. Those beetles are just out on a mushroom because that's where they live their whole life.
Yeah.
Besides under a piece of wood when they're estivating. Yeah.
I did have a question about it. I forgot to actually get to, which was one of the things I do try to do too, is think about the size, say, of a bracket.
Right.
Are there any associations that you know about where you feel like those older, larger perennial polypores, like that they need to reach a certain age before they become good habitat, or is that like open?
Well, I would think that that is like an idea that could be expanded into a bigger concept. We see that a lot with other species where it's old growth, right? Like there's like other resources that the old growth supplies as it gets more developed. And so perhaps like you might provide by leaving the Ganoderma applanatum, the little beetles that we saw in there might have a population big enough that when something new moves in that eats them, we now have a dynamic that's new, and like it invites another thing and invites another thing. Like the dynamics of a forest take a lot of time. And even my backyard is really interesting to me in that concept where you can see in our trees someone tried to do a timber sale and then it didn't happen, probably because the topography is really bad, but our neighbor's property got logged and the forests are so different. And in our yard, we have salamanders that are associated with old growth. They don't. Like, so it's just something that like even within the span of like 80 years, which seems long to us, but not long to ecology, can make a big difference that supplies enough leaf litter for something to live in, to hold enough moisture for a fungus to survive long enough, right? Like the accumulation of leaf litter and moisture and soil formation could be the defining thing for a fungus to show up for, and then for that insect to finally find that fungus. It could be years, right? Super interesting thing. I wish we could live forever and not make data centers.
I know that's the natural, like the very like naturalist or biologist feeling, like not only wanting to protect the habitat, but also like I wish we had enough time to observe every beetle all day.
Right.
Yeah. Any other questions people have, please feel free to raise your hand, unmute yourself, and just ask. Put them in the chat. And I may or may not be able to see you if you're raising your hand just on the screen.
Oh, Brennan, you said there's a secret stand of mature chestnut near La Crosse.
Mm-hmm.
Yeah, is that where you went to visit?
Yeah, but it's actually gone.
It is.
I was at a bar. This is crazy. I was at a bar last year in Delaware. And some dude started talking to me. We showed up all muddy, like I was doing work. We showed up all muddy. He starts talking to me, and I was like, oh, yeah, we're plant pathologists. Like, I work on chestnut blight, this thing, because people usually know what that is. And he was like, oh, I'm from La Crosse. There used to be this big, like, forest there of chestnuts. And I was like, that's where I go to work. And, like, but the professors, and this goes back with wanting to live forever, the professors that were associated with that research retired. So no one applied for the grant, no one applied for the research, and it ended. And they cut down the trees because the property, and like I think the property owner
yeah, it was a little bit more complicated. I don't necessarily know all the details, but I know at least one of the professors involved here, Dr. Anita DeBello, she's definitely still working at UWL, and her funding didn't really run out, but I think it was the loss of that habitat. It was the property.
Bill McDonald at West Virginia University.
Oh, who was the other person associated that might have had the funding? Yeah, one part of it was this was kind of she told it as a cautionary tale about having your research be on lands that are like private,
right?
You know, and you're trying to stay in contact with the private landowner, and they can at any time be like, "Well, I just wanted to clear all the trees."
Right, I know, and I was like, I heard this at a freaking bar from some dude, and he was a pilot, like, and so he wasn't even like a scientist; he was like the neighbor of the dude, and I was like, what the fuck? Yeah, so crazy. So I learned about that site in La Crosse doesn't exist anymore, but we used to go there, and someone put Inception in here, organism on an organism. So I used to work with a virus that infected Cryphonectria parasitic of the fungus that infected American chestnut. And so that site, we would go out every year and we would take the fungus infected with the virus. We use the analogy, it was a lot like AIDS, like it really like immunocompromised the fungus, and then it would spread, because they sexually mate, like they would mate and they would spread the virus that way. And yeah, it worked, but it also was a really interesting experiment where it was like, it worked over 40 years. So it wasn't deemed as like a thing that someone could use to save the American chestnuts, because it was like decades long. until you got results.
Yeah.
But it was cool. It was cool to see big old American chestnuts. It was nuts.
Yeah, I still
Yeah,
have.
it's been disjunct for like what was like many decades, I think.
Yeah.
Yeah.
Oh, yeah. That's why you guys had it and it was protected because American chestnuts were planted in Wisconsin, not out of Appalachia, because they were, you know, what people made their houses out of, made their coffins, their cradles, their food supply out of, fed their animals. Yeah. Interesting stuff. American history.
It is. I have a question from Beth, who is also a beetle woman, we'll say.
Beetle woman.
Yeah. She says, fantastic talk. I was excited to attend. Thank you, Kristen. Are you attending NAMA UP this year? So that's the North American Mycological Association's annual foray, and it's going to be in the UP. So this means, like, are we going to see you?
Whoa. I actually have always wanted to go there, and I don't know. I don't know what the date is. Is it like September? Isn't that normally when it is?
August. Somebody correct me if I'm wrong, but isn't it like 27th to the 29th?
Oh, to the 31st.
27th to the 31st.
I don't have any plans to go, but I could. The semester starts in middle of August, but who knows? Why not?
It's the Friday and the Saturday that are like the big, like
The
most
big
of the
days.
We'll also just plug the fact that Beth Yanis of our club is doing a ambrosia beetle talk.
Oh, nice.
Yeah, we are the now beetle crazed / fungus crazed.
Oh, that's great. There's a Facebook page, which Beth probably knows about, called Frass and Noodles.
Brass and noodles.
Brass. Capital Y-E-S, yes. Yeah, frass is Frass. That is the poop of the beetle, the ambrosia beetles, or any insect. And when ambrosia beetles are digging into a tree, imagine their head going through and they push the poop underneath them and they compact it as they continue to go into the tree. And so it makes sawdust, like toothpicks come out of the tree, and they cutely call them noodles.
Some contact. There was a question from Glenn too.
Hello, Glenn.
He has asked, I have been led to understand that beetles specialize in the hard polypores because they have mouth parts to excavate or eat them, while flies specialize in the fleshy fungi because they have the quick reproductive cycle. How much does that explain?
That sounds right to me. Like, that sounds good. The thing about all living organisms, and especially insects, because they're very, very diverse, is that they don't have like a rule. Flies do reproduce pretty quickly, but like there's crane flies that live as a larva for years in the water. So there's always an outlier. And if you think about it, even the polyporous beetles that are doing it, like the Ganoderma, that's pretty quick, right? Their whole life cycle is within May to September. So, I mean, I think that's like a really good broad idea, but it's not like a yes or no. But yeah, I agree with what you put for the most part. There's always a weirdo that didn't read the textbook we write about them.
That's so true, and it's true of fungi as well. We often say they don't read our books.
Yes, I know, it's cool. I'm excited. The morels are up. Do you guys have them yet? Is it too cold still?
No. In little pockets, we have little like micro habitats where the first one of the year is almost always found by Shannon Pump in the Green Bay area, and it's like an isolated little blip, and that's already happened. So that's one of our harbingers. I think it happened last week.
Oh, cool. Very cool.
And then I've heard of some people in southern Wisconsin finding them, and I see on iNaturalist people like right on the border to us have them. So I would say people start getting out and looking now, especially maybe in those like conifer mixed woods for the black morels.
Oh, cool. That is neat. Yeah, I've always wanted to go up to the UP. I was supposed to go to the God, I know it's Nicolette. Yes, I was supposed to go there and then the world ended.
Yeah.
So I have never been up to that part of the forest, but I have done, like I said, I was in La Crosse for the chestnut stuff. I've been to Winona, I've been to Chippewa Falls. And oh, the Dells. Yes, the Wisconsin Dells. I've been there for oak wilt surveys, but I would love to get to that northern part. And I was supposed to go up there for oak wilt surveys because you don't have oak wilt like big pockets or widespread up there. And we were going to do like a survey, but that was deemed unimportant and they fired everybody. So there we go. We'll be
Yeah,
okay.
we'll be fine.
Yeah.
Yeah. Well, I will ask, yeah, that's the feeling, and I'll ask Glenn's other question here. Besides Genaduba suge, how many fungivores are obligate to specific species? Oh, that's a cool question. Yeah. Like, do we know a lot about those tight, like obligate, like they have to be with this fungus to this beetle?
I don't know that, but I'm sure that there are reports of specific species that that are obligate.
Yeah.
But I mean, this whole group of these beetles that are like 2 millimeters large, like I feel even now, just with those like clips you put on your iPhone and like the citizen science and the internet, how many entomologists were there beforehand? And even when I introduced myself as a plant pathologist that works on fungi and forests, there's like 5 of us. There's 5 of us. And then there's like all these plant pathologists that work on soy. There's thousands of them. So I don't think there's a lot of reports about it just because it's so obscure and like it wasn't associated with money. But now we have the chance to inspire ourselves and our friends to get out there and try to make observations and figure it out.
Yeah, and even those observations where you can take a picture of that beetle and get it to family, that's still useful information, or you can get it and we can get it to genus, that's still useful is what I would say.
Right.
I do wonder actually, ask this question real quick, and then I'll get Marine's. So with fungi, we have a lot of sequencing projects. Like
Yes.
we have citizen science sequencing projects. Is there anything like that for beetles? Like
there is. Yes. And so I think I was trying to go fast on my slides. I'm not going to pull it up. It's okay. But like those ambrosia beetles, like they have like week-long events and that they often post on Frass and Noodles where people learn how to identify them morphologically. But my previous lab with the Forest Service, we were actually extracting DNA and identifying subspecies and species of beetles with DNA. And I think that is also something that is like scientifically relevant now, where it maybe costs like $1,000 for us to do 60 samples. That's nothing. Like I know $1,000 is a lot for us to pay our bills at home, but like with the government and universities, $1,000 is like they just shred it and like throw it in the toilet. So if you like have the resources now to identify things, it's a lot easier. And there were people looking at that. And a lot of universities, like University of Florida has a really big ambrosia beetle graduate school body. Like they have like 6 students working on just bark beetles, trying to identify them and identify their genetics, but also identifying like what those genetics mean, which we kind of do have the ability to do now. That's like the next tier of technology that is inhibitory because of the cost. And so I have done that before where you can identify the proteins that are responsible for doing something, like what gene in your body makes it that you can digest milk. Like we know that with humans, but it's a lot harder to do with plants and bugs. But it was really, really expensive. And it was like you had to have like, it was like I had to read a manual that was like this big and like I had to like literally blast. I had to make a liquid and shoot the DNA with lasers and it exploded and then I measured the molecular weight. Like it's crazy stuff. So you need like a super crazy skill set to do that, to identify proteins, to figure out what they do. But right now we're at the point where like people like you can like go to an afternoon and learn how to extract DNA and then send it to a lab. Like that's amazing. So I think that there are a lot of efforts that are happening and maybe like the Coleopteran Society is doing that, just like you guys as the Wisconsin Mushroom Club are doing that.
Cool. We'll have to get connected up. I'm going to ask Maureen's question now, which is a really good question. What beetle mushroom work are you excited to be working on next?
So I, again, work on fungi that grow in Petri plates and don't make mushrooms. But the thing that I got excited about, and I am excited about, and I just have to figure out when I'm going to do it, maybe this summer. So a lot of those beetles that carry the fungi on their body, right? They transport the fungi, and the fungi is a pathogen of a tree species. Well, I have always wondered what would happen if I want to know what the susceptibility range of those pathogens are. Often they're very specific because they have the DNA that allows them to break down this type of wood, whatever. And they're very constricted when they're in those symbiotic relationships. But beetles don't see very well. They smell. They go in a direction based off of smell. And so there's a very high probability that they're flying towards this tree and they land on this tree, which is a different species, and they will drill into it and they'll put their fungus into it. And so when I'm working on controlling Tree of Heaven with a beetle that carries a pathogenic fungus, I worry and I'm interested in knowing what will happen if a beetle doesn't see right and goes to a black locust and infects that tree with a black, like with the pathogen. And I have looked at that a little bit in doing like host range studies of the fungus, but I haven't tested everything. What if that beetle goes to like an obscure thing that I wasn't paid money to look at because no one cares about it, right? Like, I don't know, fire cherry, right? A really regional like tree that no one cares about. Like, what if like that pathogen could kill fire cherry? So that's what I'm interested in, knowing the genetic potential of a pathogen going to a different host because a beetle accidentally sees the wrong tree. That's what I'm interested in. Sorry, it didn't involve a real mushroom.
Well, that's okay. And I'm sure there's some that produce a real mushroom from time to time. And we love all fun here in the
right.
Like
in my field as a forest pathologist, armillaria is the #1 mushroom producer that is a pathogen.
Yeah. And some of them, I mean, like, and that's a delicious one to us.
Right. Yeah, I eat it. We have a couple in the backyard, a couple species in the backyard.
Okay, so Charlotte had a question. Let's see. Have there been any cases of insects actually threatening specific species of fungi, or are they in pretty good balance? That's a really interesting question, like invasive, maybe.
That is a super interesting question, and I don't know. But what makes me think about your question makes me think about, what about the inverse? Like, what if we found a beetle that wanted to eat golden oyster and could like biocontrol golden oyster? Because that is what like my job is, like to find things that already exist so we don't have to use herbicide or pesticide to control things we don't want.
I will say, this leads me to a quick question I'll ask. I always feel so threatened by gut biocontrols, as I'm sure working in the field, you know the dynamic where you're like, yes, we think this can control this, but actually it ends up eating all of our oysters. Like, I'm assuming that the process of selection is really long.
Yes. So the thing I'm working on now, which is almost ready to go to be made into a product, it has been studied for 25 years, like by literally hundreds of people. And like there are laws and there are rules, and like sure, the example exists of the ladybug being introduced. That was before we knew anything. Again, we have to make mistakes in order to learn how to not make mistakes. And so biocontrols are really serious now, even with something that works really, really well. I love that emoji that's floating through the crowd. Even something that works like really well, like Silver Bullet, like there are like people who are just like, no, we're not putting that out there. We're not taking the chance. But instead, they just use a bunch of pesticides. So
yeah, that's another one of those balancing acts. I partly, bring that up because I know sometimes people still buy the ladybugs as biocontrol. Like,
yes, I know. Yeah.
And that is one of those things where it's like, the popular market versus where scientists are at right now is not the same. So, Ananda had a really cool question that Jessica Peters has put an answer.
Yes, Jessica answered it. Thank you.
But the question is, do we know what spreads chaga? So that is the much prized sclerotium of Enotus obliquus. And it goes to like mostly birch trees, but I've seen it on oak once with a friend.
Oh yeah, that's right. Yeah, so that's the correct answer. It does make spores, even though it's called sometimes sterile conch. That's like a really old common name of Enotus obliquus. And actually there was a really big thing, again, legal stuff. This is always building. These conversations always build. But when I was with the Forest Service, we actually did a survey and we did some we actually made like legal laws for people who wanted to grow chaga as like a forest product. They wanted to take plugs, like what you would do with shiitake. They wanted to take infected chaga plugs and they wanted to put it into birch trees and grow chaga to sell. And the Forest Service said no. And like legal, like APHIS and like imports said no, because normally chaga happens at like 3%. That's why it's so rare and prized, right? You don't find it. Incidence is like 3%. And so there's like birch genetics that are matched up with chaga that allow it to happen. Like it's weak genetics in that way. And if you take a plug and put it into a birch tree, you're kind of like bypassing like the natural immune system of the tree. And like you could wipe out birch trees by making this market and increasing, augmenting the population of chaga. So yeah, it's spread by spores, and it also has to be, it's like a really weird life stage of the birch, that it like, it's a seasonal thing, and then it's also like a health thing of the birch tree. So it's like, that's why it's so rare. Right. Yeah, and the spores are also like under the bark, and they only make the spores when the birch tree dies, right? So the birch tree is alive when chaga, and then like if the birch tree dies eventually, then it makes spores, and that's why people think it made spores for so long, because they just didn't find it, because the birch has to die, and it might live with this infection just like you live with the athlete's foot lives with this infection for so long.
Yeah, and that's one of, I was adding that as like some context for the chat, that like there's two different structures we're talking about here. We got the sclerotium,
Right.
the chaga that we like for the tea and such like, and that is sterile. But then it seems to be maybe where the fungus is storing stuff for later, basically. But then when it dies, it creates this like sheer, beautiful. If you get a chance to look under dead birch bark, and if you do, please take pictures or like video. It's a shiny, kind of iridescent looking polypore surface that you can see where it does make its spores once the tree's dead, like you say. And yeah, I'm gonna follow up with Ananda's comment really quick here, because she says, I remember Britt talking about the beetles in the decomposition cycle of chaga, I think, like of the sclerotium maybe.
Cool.
Which is cool. We should all look for that. But when you're harvesting chaga, this is also because it's on live trees, just another ecological note. That's why we like the hammer method, where you're like chipping off instead of like trying to saw. The tree's still alive and it could stay alive for a while.
And make more chaga, because it has the genetics to support an infection.
Exactly. You want that susceptible birch tree to keep chugging for like another 15 years or whatever. Jessica said it's so interesting and kind of like the sustainability for chaga. Yeah, that too.
Yeah.
Glenn has a question, which we can wrap it up whenever you want to say. No, I'm just continuing to ask.
Sure. Let's take that last question, and then I'm gonna go to bed.
Okay.
It's 9:30 for me.
Oh no.
But I had no, it's fine. I had a ton of fun, and I can sleep in tomorrow. I'm excited.
Okay, so Glenn asks, do you have any thoughts on how we can build a case for protecting old growth forests of types? For example, wet forests are less likely to be a source of raging hot fires that you're concerned about. And I guess maybe also connecting to beetles and beetle life.
No, that's fine. I don't know how to get people to, I mean, there is like kind of a big energy associated with old growth right now, just like obviously you have, where people are caring, but it's, I mean, that's going to get into a political conversation, right? Like who has more power and pressure, right? Someone who wants to build something on a location that happens to have old growth. But yes, awareness and showing people how important they are and the biodiversity they support. And just like Mariah put in the chat, the Old Growth Forest Network is an active group that is trying to save properties, and they don't, it's hard because a nonprofit like that has to do so much in order to like buy properties and to continue to manage them, because even though you have an old growth forest, it doesn't mean it's not susceptible to being invaded by multiflora rose or other invasives. So it needs active management, and it's really, really hard. But I would say, it's maybe it's not the answer you want, but like take a loved one to an old growth forest and show them how important it is. Like awareness is a really big deal. You never know where that person will be with like what influence or power they might have to protect something you care about.
Yeah, spread the love of it. Take them to the woods and get them hyped on fungi and beetles, is I think. Yeah. Oh, and Beth wanted to note, just real quick, tree variety mentioned the University of Florida's pitch tube page mentioned these very issues. That's cool. Follow up with that. Perhaps when we post this video, we'll put a link to that. And yeah, this is probably your bedtime, so I wanted to say thank you so much, Kristen. This was the coolest, as Jeremy says in the chat.
Thank you. That's kind of you.
Yeah, we love these interactions, and it adds depth. Also, I live in La Crosse right now. If you're visiting, if you need to come here.
Oh, that's nice. Thank you.
Stay with me.
I did scribble all of this stuff down, and it is possible that I could maybe come to NAMA. I do enjoy NAMA a lot. It's very, very cool. Yeah, so thank you for telling me about that. It wasn't even on my radar. And I mean, it is just a weekend, so I could probably make it work. And it's the beginning of the semester. I'll just let them read a paper.
Exactly. Feel like professor's on vacation. Good that.
Oh, God. Yeah. All right. Well, thank you, everybody. It was really nice to meet all of you. This was very fun. You have such great questions. Thank you for paying attention.